From Microbe TV, this is TWiP, This Week in Parasitism, episode 118, recorded on October 4th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. This episode is also sponsored by Drobo, a family of safe, expandable, yet simple-to-use storage arrays. Drobos are designed to protect your important data forever. Visit drobo.com to learn more. Hi, everybody. I'm Vincent Racaniello, and joining me today here in New York City, Dickson Despommier.
Hello, everybody.
And Daniel Griffin.
Hello, everybody.
And the weather has returned to
It has taken
late
a turn
summer.
for the better.
We have nice skies today with clouds.
Beautiful.
We did get some rain last week, but now the grass was sufficiently dry that I could cut it yesterday or Sunday.
Nice.
Today is a beautiful day. You look out there, it's that just beautiful blue and the clouds.
It's like a...
It's very nice.
Yeah, it's a picture-perfect day.
We have we're recording a few days early. Right?
We are.
Wednesday, 2 days.
Yeah, but who's counting?
So it may catch a few listeners unaware, yet Daniel has other issues that he needs to deal with, so we're going right ahead.
Don't we all? I don't broadcast my issues, but I have some issues.
You don't have to broadcast them; they're just oozing out of
Actually,
your
we're
pores.
broadcasting right now.
No, I think we were gonna record tomorrow, but I have a couple people coming in for interviews to join us in the lab. So
yeah, exactly right. And thank goodness we're not tomorrow because I have to be somewhere else.
Are you going to Bolivia?
No, I'm just.
What's the principal export of Bolivia?
Phosphates, I believe.
Tin. When I was in school, I just remember that from history. I don't remember anything else. Maybe social studies. That's the only thing I remember. What is the principal export of Bolivia?
What course duld they teach that in?
I think it was social studies.
Social studies?
Social studies.
Or maybe geography. I don't know.
Was not lost on you.
Tin. I would who cares?
Right.
Apparently you do very much. You've maintained that knowledge.
Romans
the principal export of any country is viruses.
Yes.
All right, let's go over our TWiP 117 case, Daniel.
Okay. I'm trying something new this time. Usually I keep my own notes and use those, but I'm going to just strictly go with your notes, Vincent. So
I try and type as you speak.
Yeah. So I've been noticing, I'm saying, why am I doing all this work?
Yep.
Vincent's doing such a good job. So as I like to say, for those of you tuning back in and for those of you tuning in for the first time, our case from last TWiP was a 66-year-old woman who was born in Guinea, Africa. She grew up there, but then moved to the United States about 10 years ago, where she worked until she just retired. She lives in Washington Heights. That's this northern area of Manhattan where we are. And she reports that her mother back in Guinea was having some eye issues when it was going to need to actually have a surgery. So she went back with her 2 daughters, was there for 3 months in 2016. She actually just got back. This is a recent case. Came back about a week before she started to have some issues. What were these issues? She was reporting a headache, recurrent fevers. She was feeling poorly. She says that when she was over there, she was staying in the big city with her mom. Mom's in her 80s. She actually said she had very nice accommodations there. In the home, she had her own room with her own private toilet, and it was a nice home in the city. But they actually seemed a little offended when I brought up screens or bed nets. They said we didn't have those. Maybe we'll even talk about that later on.
Need them? She said she just
didn't have them. She had been married for 40 years, but recently had divorced. She's had a number, did I say 10 children? That many children. Yep. She's currently not sexually active.
She had enough in her lifetime.
I beg your pardon.
10 kids.
Stay active. That
10.
doesn't mean you have to have children.
10 is enough.
You could be active and not have any kids.
Just kidding. I'm just kidding.
Okay. Just want to
Who am I
clarify.
to judge what someone else
Exactly.
Okay. So she said that this began with high fever, and the fever would last several hours, and then it would break. She'd feel better, and then 2 or 3 days later have another episode, right? So this started a week ago, and now we're about 2 weeks later. So she's been having these high fevers every 2 to 3 days. With the fever comes blurry vision, double vision, headache. She initially went to another hospital, right? I don't even know if I mentioned the other hospital. She went there with these complaints, and they sent her out of the ER. So then she comes to Colombia. And at the Colombia, they go through the Ebola screening questions. We still do those for anyone from sub-Saharan West Africa. They were all negative. She says she had no diarrhea, no urinary discomfort. Her back does hurt. And that's sort of an interesting thing that I'll have to say people with this complaint often report. She feels that her mouth is dry, and she ends up getting admitted here to Colombia. As far as past medical history, she has high blood pressure, cholesterol, diabetes. She's not overweight. Her appendix was removed. There's some unknown reaction to Novocaine in the past. Doesn't smoke, doesn't drink. And on physical exam, she has a elevated temperature to 39.4. It's well over 100 for those using Fahrenheit. She's breathing 14 to 16, so slightly more rapidly than normal, but not a lot more. Heart rate is over 100, so she does have a rapid heartbeat. She does have a 2 out of 6 systolic murmur that's radiating to the left carotid. We characterize this as a high-flow murmur. No jugular venous distension. She doesn't appear to be volume overloaded. Her abdomen, she has a slightly enlarged liver that's not tender, and I'm actually able to palpate a spleen tip in the left upper quadrant. So I'm saying she's got a slightly enlarged spleen, as well as a little change, I'll say, in consistency, right? So it's palpable now. Normal bowel sounds, no rash. She has an elevated white count with bands, 9% bands, 0.1% eosinophils. Her platelets are down at 79. Little anemic with a hemoglobin of 11. Bilirubin 1.5, bicarbonate 20. She has a chest x-ray that is clear. Slightly small red cells. And I think we threw in that she did not like animals. We were asked about exposures.
Right.
And there was no animals. Not only were they not exposed to animals, but they don't even like animals. It was the middle of the rainy season back there in Africa, in that region of Africa. Apparently she loves to walk outside in the rain. Her daughters who were there when I was seeing this woman were quite entertained. The fact that whenever it rains, she likes to walk around in the warm rain. No cats to keep away the rodents. The food is all prepared in the home, and she eats all her favorite foods. But then I pressed, what were these favorite foods? It was rice. Apparently she eats lots of rice. No sick contacts, no exposure to the healthcare setting. That was the Ebola screening stuff. And no bug bites, at least that she reports.
Dan, just as a side, if someone comes from Western Africa and you do the Ebola screening and they answer yes to any of them, what would you do differently?
Then we would actually do testing, and you would put them in basically a barrier situation where you're not exposing the healthcare workers.
So if there's no potential exposure in West Africa, you would not do any Ebola tests, right?
True.
Daniel, take that first guess. We only had a few, but...
All right, I'll do Yosef. Yosef writes, Dear TWiP team, one thing that I want to mention is that we are taught in our medical school that we should always ask our patients what they think is causing their problem. I love that. Did I mention that?
Yeah,
last
we
time?
did.
You did, actually.
You did.
I do ask my patients occasionally, but the most common answer I get is either I don't know or isn't that why you are here? I find this question to be really helpful for another reason, allaying a patient's fears. I commonly get a response that they are concerned their malady is a cancer or another diagnosis with significant morbidity. And being able to calm a patient down with an explanation of what may be going on is an amazing way of promoting the doctor-patient relationship. For the case today, my guess would be malaria. To be more specific, I would say it's probably Plasmodium vivax, since her fever seems to be recurring every other day. You should ask the patient if she could pose for a textbook since she fits the case so well. Splenomegaly, anemia, thrombocytopenia, headache, and fever all point to malaria. I would assume that since she has had malaria before, she would be able to say if it feels similar to that event if she were to be asked. I would get a peripheral smear to confirm. I would get an ova and parasites.
I would not
get an ova and parasites or an original video animation. Okay. I would treat with chloroquine, but if the patient does not get better within 3 days, or I may find out that the strain is resistant, then I would use artemisinin combination therapy. Sincerely, Yosef Davidoff.
Dickson Despommier.
Yes. Wink writes, I think our traveler to Africa with periodic fever is infected with Spirillum minus and has rat bite relapsing fever. This is based on clues Daniel hinted. On further reflection, this show is TWiP, not TWIM. So I'm going to have to say malaria. But Daniel likes to keep us guessing, so I'll go back to the bacteria, S. minus. But maybe he's throwing a curveball that's still a parasite. So I'll say P. malariae or P. ovale. After further thought, I'm finally going to settle on a form frusta of Ebola.
What's a form
Wink
frust?
Weinberg.
Do you know what that is, Daniel?
I don't know. What is a form frust, Chewy?
Form frust.
I'm going to look it up. Form frust is atypical or attenuated manifestation of a disease. So
Interesting.
attenuated Ebola. Interesting. Well, Wink is a little equivocal.
Well, Wink has a lot of good thoughts,
Yeah, that's right.
and I appreciate several of them.
We have one more from Adam, who writes, Dear TWiPinella,
TWiPinella.
I started listening a couple of months ago after a friend recommended TWiP and am now finally up to date. Firstly, I shall address the case study. My guess is malaria, either P. ovale or falciparum. Given the time between fevers and their co-endemicity in Guinea, perhaps the former species, if the lady does not feel like she is dying. Admittedly, diagnostics is not my strong point as I work more on the molecular side of parasitology. I became interested in parasites after learning about guinea worm in secondary school and geography lessons, surprisingly. After studying biochemistry at the University of York with a stint at the University of Valencia and learning more about NTDs, I am now working on my PhD at Queen's University Belfast.
Nice.
Under the guidance of Mark Robinson, one of the co-authors of the NOD paper from TWiP 82.
How about that?
My research focuses on the extracellular vesicles of helminths and how they are made by the parasite, work that I am very much enjoying despite discovering the less glamorous side of parasitology during abattoir visit.
You're right.
That's if a glamorous one exists. I wouldn't want to blow my lab's own trumpet, and yet I am yet to publish myself, but there is currently a lot of interesting work going on around the extracellular vesicles of both helminths and protozoa. Perhaps I could recommend the following paper for discussion on TWiP on the extracellular vesicles of Trypanosoma brucei, entitled Extracellular Vesicles from T. brucei Mediate Virulence Factor Transfer and Cause Host Anemia, if anything, just because it has nice videos. All right, we'll do it in a future episode. What do you think?
Absolutely.
Also have a question. Among Dickson's many repeated anecdotes and nuggets of information, an association between helminth infections, possibly Ascaris lumbricoides, and Clostridium difficile has been mentioned at least twice. Would he be able to provide a source of information on this? It interests a friend who studies C. diff, but we have been unable to find anything in the literature bar a mention in pigs. Additionally, Dickson mentioned the Chiclero ulcer, leishmania from rubber tappers in Latin America. I would like to add that I remember being told in lectures about shifts in society causing the Chiclero ulcer to now also be associated with human drug mules who venture into the depths of the jungle to avoid detection while smuggling. I really appreciate all the time the three of you have put in, primarily in combating polio and NTDs, but also in producing TWiP. Sometimes when looking out over a gray Belfast on a particularly miserable day while listening to your podcast, I think, at least I don't have ... Adam is a PhD student at Queen's University Belfast. So, Dickson, you know anything about... Do you have a reference for this C. diff worm?
I actually don't, but I do know that enormous boluses of Ascaris in small children cause bowel stasis. And anything that will cause bowel stasis could ultimately lead to C. difficile, because C. difficile is an anaerobe. And once the aerobic environment of the small intestine goes down below a certain level, the spores of C. difficile turn into bacteria. So I'll look hard for that and get back to you.
How can it be that there's no literature?
Well, I'm not sure, but maybe Peter Hotez might have an answer for us.
Maybe you made it up.
I didn't know. No,
Daniel, does this ring a bell with you?
You know, it's interesting.
Does this ring a Belfast with you?
It's interesting because if you think about the epidemiology, C. diff, Clostridium difficile, is a major issue here in the developed world.
Sure.
And it's actually, I think, currently one of maybe actually the number one nosocomial infection that we struggle with in the hospital. Where Ascaris, you think of more as a first world, as a developing world problem. So yeah, it'd be interesting. I don't know. I don't know what the dynamic would be there.
Yeah, it was a mere speculation on my part, but I think based on the fact that a lot of children do die from Ascaris without an explanation. It's possible that this might be the underlying cause rather than aberrant migration to some other part of the body.
Yeah, there's an interesting issue between C. diff and age. If you look at children under the age of 2, they developmentally, they're actually not vulnerable to C. diff yet. They actually have a lot of C. diff, but the chloride channels are involved in the toxin. They're not vulnerable to. So as they get older, let's say 5 or 6, then you can actually start seeing a real C. diff colitis. But it is interesting. There's a high incidence of C. diff that lives in the young colon, in the under 2 years, but without the toxigenic effect. So
interesting.
All right. So we had Plasmodium ovale, falciparum, malariae, and vivax, and spirillum minus.
Exactly.
Those are the guesses.
No one went with Lassa. I thought we were trying to push that there, Lassa fever, but
nope. No one.
Should we try to guess?
Well, yeah. Why don't? Yeah. Should we? Who wants to go first? One of you guys?
Well, I was thinking of a malaria myself because of the recurring relapsing fever.
Yeah,
right?
but which one?
I don't know. I'm not that good.
You're terrific, but at other things.
What?
So the possibilities are, if it's going to relapse, if this woman has had a history of malaria at some time in her past, because that's where she's from. So her state of immunity is not 100%. But there is a memory there, so that when they do encounter it again after being away from it for some time, they're susceptible, but they're not as sick as if it was a new infection for the first time. This is what I've been told by people of great knowledge in the malariology field. So the guess as to which kind of malaria she might have relates very much to as whether she's Duffy blood group positive or negative. And there is a large group of people living in West Africa that are Duffy blood group negative, and so therefore they're not susceptible to P. vivax, which is a relapsing malaria. But ovale, which is also related to vivax in some remote way, also relapses. So it sort of takes the place of Plasmodium vivax in that area of the world. So I would guess ovale. But it would be interesting to see the blood
it's interesting when you go to the CDC and you look at the incidence of different malaria species. They have a drop-down menu. You can pick your country. If you pick Guinea, the predominant malaria species is
Plasmodium ovale.
No, falciparum.
Oh well,
85%.
Of course.
Ovale is 5 to 10%.
Okay,
which she could fall into that.
That's true. But if she had falciparum, she'd be a lot sicker, I think, than she was.
Vivax is very rare. In Guinea.
That's exactly right, because of the Duffy blood group factor being so prevalently absent. But we would need to see some either molecular evidence like a PCR, which would be specific for each of the species, or a blood smear, which you can identify the morphology.
You don't want an original video animation?
only if it's produced by a medical group that's very knowledgeable.
All right.
Well, what do you make of the fevers, right? We learn, like we teach a lot about the different fevers. And so why don't we let you speak to that? What are these different?
Sure. So there's quartan malaria, and that's fever every 4th day, but it's actually every 3rd day because you have to count the first day as day 1, the second day is day 2, the 3rd day is day 3, the first day again is day 4, actually. So that kind of screws things up, because I think the Greeks didn't know too much about counting, and they were the ones that named all of this. And then there's tertian malaria, which has fevers every 3 days. And this is every 2 to 3 days. So I would say. Maybe she wasn't keeping track, or maybe her level of immunity modified the periodicity of her fevers, and it wasn't exactly a synchronous infection that she had. So it could have been for that reason that that you weren't seeing such a pronounced synchronicity of her fevers.
Yeah, it's really interesting.
That's my guess. That's a guess.
When people come back, say the expats or travelers, they come back, as you're discussing this synchronicity, the synchronicity takes a while to develop. You've usually had to have malaria for a while for the parasites to get in this sync where it's every, let's say, 48 or 72. And then, I think as described, different malarias have different periods of time. But I can't say I would ever use that, I guess, personally to say, oh, this is what you have, I'm going to treat you.
Exactly.
Our default is to assume it's the worst malaria, which would be the falciparum.
That's right.
And the majority of deaths are so we usually assume there, start to treat there until we're sort of proven that it's something else. And then, doing the blood smear, I think, is something we talked about, to look at the blood smear to see if we can figure out what it was. And it is kind of, as you mentioned there, Vincent, it's a numbers game. She's from an area where it's not going to be Vivax, pretty much. No. It's not going to be Nolesi, right?
Knowles-eye. It won't be Knowles-eye.
Won't be Knowles-eye. Let's call it Knowles-eye because the guy's name was John Knowles, so we'll actually pronounce his name correctly.
I've heard it said that way, too.
Yeah, so we'll call it John Knowles-eye.
Right.
So it's not Knowles-eye, it's not Vivax, but we're left with, could it be falciparum or falciparum?
Yep.
We'll go with falciparum.
Either or.
Could it be ovale?
And the answer to that question is yes.
Or could it be
both?
Could it be malariae or could it be mixed?
Could be mixed. She could have a mixed infection.
Yeah.
She was there for 3 months, so that's enough time for different species to get involved. And I've seen
her during her walks in the rain.
Yeah, right, exactly right.
Or her open windows with no screens.
Yeah. We don't need mosquito netting.
When would they bite you? What part of the day would they be biting?
Evening.
So the walks in the rains are not a risk factor.
Well, she was there at night, didn't she? Said she.
Oh, she might have walked in the rain at night.
Yeah. You don't know because I think when you get the urge to walk, you walk.
Yeah. No, the anopheles is interesting enough. It is, as you mentioned, it's usually evening, nighttime. They're not day biters like we worry about with those Zika mosquitoes, right? Mm-hmm. And they like to sort of bounce around, right? They like to sit on the wall. They like to then come over and have their blood meal. And, so, when I was worried about malaria recently when I was in Asia, and there was nowhere to hang a bed net above me. So how am I supposed to use my bed net? So what I ended up doing was I draped it on the wall. Uh-huh. And the idea that then as the anopheles were bouncing around, they would bounce there. It was insecticide treated, and then they would die. So in this case, the other thing that I really liked about this was that the very clearly palpable spleen. So, I mean, for me, this was sort of a slam dunk diagnosis. Here we have a woman, 3 months in Africa during the rainy season, taking nothing to protect her from malaria, comes back now with a periodic high fever with shaking and a palpable spleen. Mm-hmm. And we do the blood smear, and yes, we see malaria on the blood smear. 2% parasitemia.
Ooh, that's pretty high.
Now, the 2% bothers me. And actually, I said to the team, like, as we're seeing it, I'm like, why 2%? 2% bothers me because 2%, it's difficult for me to know what type of malaria she has. And so I explained this thing, is that, so your Vivax and Ovale like to infect your young erythrocytes, which are going to be about 1% to 2% of your red cells. Huh. So at 2%, it could be that.
It could be 100%
your malaria, plasmodium malaria, like to infect the older, again, that could be 1% to 2%. Your P. falciparum can infect any ones they want. Exactly. So that could range from 2% all the way up to much higher. So at 2%, I still don't really know yet. Right. I mean, I almost want it to be 4% so I know it's falciparum. Yeah. Or,
So how is that determined? And I can actually speak to that issue because
answer your own question.
Exactly, it's a rhetorical issue here. You take a blood smear and you make a thin smear and a thick smear. Thick smear is done just so to concentrate the red cells, and you lake open the blood, so you get the hemoglobin to leach out of the red cells. So now they're all ghosts. Then you stain with Giemsa for both the thin and the thick, and you look to see if you see any parasites on the thick smear, and on the thin smear, you use it to identify the morphology. Okay. So you are looking in oil. All right, so that's like 100x on a microscope. And within that field, if all the red cells are touching each other, but they're not on top of each other, they're just touching, there's about 200 red cells in each high-power field. So if you have 2 red cells that are infected, and you have 200 cells, that's a 1% infection. If you have 4, that's a 2% infection. But just think of what's going to happen the next cycle, right? It goes from 4 to 8, and then from 8 to 16, and then from 16 to 32. And the next thing you know, and within 2 weeks, you could be at death's door, basically. So this is a critical position. I mean, if you can see red cells infected in every high-power field, that's a heavy infection.
So you were able to catch her early on in her illness?
Well, so that was the thing we were wondering about, right? So we didn't treat her because I wanted to see if she was going to go to 4 and then 8. No, we treated her. No, so we did. We treated her right away.
Absolutely.
The lab was able to speciate the malaria for us in 2 ways. One was microscopically, they were able to look at the morphology and see that it had a morphology consistent with falciparum.
Really?
Also, we have a rapid test, and the rapid test is able to tell whether it's falciparum and also whether there's a non-falciparum species present as well. And so we did that, and fortunately, it was a pure falciparum infection at 2%.
And this
so they found, I'm sorry.
Rapid test is an antigen test.
It is. It's an antigen test. And there's a couple different ones. There's specific aldolases, and there's also a specific HRP. So you're looking at specific antigens that are present in the different malaria types.
So they found the banana-shaped gametocytes?
No. See, that would actually be pretty darn late, right?
Yeah.
The gametocytes.
But go ahead. This is not a latent chronic infection then. This is not a relapsing infection because falciparum doesn't relapse.
She got reinfected.
Yeah.
Which happens, right?
Yeah, exactly.
Yeah, and that's, I tell them, and I think I said to this woman as well, I said, I hate to tell you, I know you're originally from Guinea, and I know your heart is that of a woman from Guinea, but your blood is now that of a New Yorker.
Exactly.
And New Yorkers don't do well when we get malaria. So, we say that 1 or 2 years, you lose the vast majority of that immunity that you acquire while you're there. So people will come here, college student, right? They'll come and they'll be here for a year or two. They'll go back thinking it's all good. I'm going back home again. I'm not traveling to some exotic locale. I don't see a travel doc. And then they end up with malaria. And in a lot of areas, people are not using screens and bed nets. And if you were to use them, it would be considered sort of offensive and rude. Like, well, why are you doing that? What's going on? Aren't you one of us? Oh, now you're one of those New Yorkers with your sleeping under your bed net. And so it could be culturally a little bit of a challenge for them to maybe take precautions.
So how'd you treat her?
She actually ended up with a artemisinin combination pill. The co-Artam. So she was well enough that we treated her with oral therapy.
Okay.
And she did great.
How long did it take for her to recover?
So we started treating her, and by the next day, her parasitemia was almost undetectable. It was well less than 1%.
This is one of those miracle infections. I mean, I'm not kidding. We had a technician at Columbia that caught it and came back, and it smoldered for 6 months. And the reason why we know she had it is that eventually she got very sick. She was admitted to the hospital and she was treated. 3 days later, she was back at work. And she was really
miracle
sick,
drugs.
Really sick.
Miracolo.
It's like the Lazarus effect almost. You give the drug and they get up the next day and how could you have been so sick and now you're so well? And it's that kind of a multi-systemed infection.
Daniel, I presume at some point she's going to go back to Guinea.
I think so. Did
you give her any advice?
This was her 3rd time having malaria. She had it twice when she was younger. It's tough. She's not. She does not consider malaria to be a serious thing. Right?
Nobody over there does.
Why did you come to the ER then?
Because she felt sick. Like, now, if she was there in Africa, right, she would have just gone to somebody and say, "Hey, I think I've got the malaria again. Can I get some of those pills and I'll be better?"
Sure.
So, it is, I think a couple of the things to take away, as you're mentioning, it is tough for people who are returning to their home countries. Yeah. Sometimes they're returning their home countries to an area that never had malaria before, but now does. We see that in parts of India.
You know, they have the rainy season and the dry season. And if you go at the height of the dry season, it's probably they're not going to encounter it very much. And it's just at the beginning of the rainy season or at the end of it when everybody starts to come outside from their houses again where they get exposed to the mosquitoes.
Daniel, the pill that she gets in Guinea, what is that? You said, give me the pill.
So actually, interesting enough, the coartem that we discussed, there's a lot of these artemisinin combination things out there. And people, maybe they're familiar with the Z-Pack. We get like a little packet. And so the coartem and a lot of these artemisinins come in these prepackaged treatment.
Interesting.
And you basically, these are the pills you pop in the morning. Let's say it was coartem, maybe like 4 pills in the morning, 4 pills in the evening. You get like, 3-day course. And so a lot of times they have these prepackaged. We think here you go, and you take your oral tablets.
Yeah.
Dickson, unrelated question.
Sure.
Here in these parts, are the mosquito populations declining?
Oh, I don't think so.
No? Why not?
I wouldn't say
Isn't it getting cooler?
You mean as the reason of the seasons?
Here in New York.
You mean, God bless. You mean right now? At this time of the year?
It's this time of year, yeah.
The answer is yes, they are declining. But at the same time, you've got migrating birds now. Okay, this is the Atlantic flyway, so you've got all kinds of birds migrating from the north to the south, and they've got their viruses. This is not This Week in Virology, but they're carrying Eastern equine encephalitis, basically, and they're going to give it to mosquitoes along the coastline, and the mosquitoes will in turn then turn around, and after biting the bird, they have an option for biting horses, and when they do that, the horse dies. If they bite people, person dies.
Of course, you can vaccinate your horse, right?
You can, but they don't have a vaccine for people.
No. Horses are more valuable.
It's exactly what they
especially
say.
racehorses. In fact, Daniel, in your next life, you should be a horse.
I should be a race.
A thoroughbred.
I don't know if my mom still listens to these, but that was actually, she was encouraging me to be a vet, and particularly a horse vet. Apparently, she had 2 arguments for why I should become a horse vet. So the one was apparently 90% of the people at that time going into veterinary medicine were women. So I would meet a
suitable
life partner who would be a vet, which was apparently back to the first one. And apparently the racehorse veterinarians, they do really well financially.
They do really well.
And but she wasn't really,
Their big scare right now is West Nile. That's what horse owners are most afraid of.
There's a DNA vaccine, equine vaccine for West Nile.
Yeah.
Covered. They're first covered because they're valuable.
With it really fast, didn't they?
Yeah. This episode is that it for today's?
I think so. Did you have anything about this Chiclero ulcer and the human drug mule?
That's an interesting twist. I mean, these drug mules are relatively standard for high drug trafficking areas. So I have no doubt that they exist. But Chiclero's ulcer is from someone who goes out and harvests chicle from the chicle tree. So you've heard of these gums called chiclets?
Yeah.
That's where they get their name from, right?
Yeah.
So they don't get it unless they go out very early in the morning or very late in the afternoon. So I'm not sure how that figures into this drug trafficking scene unless
they
to avoid detection, they come out early and
maybe
get on their flights north or whatever. Feel sorry for those people. I really do. One added note here. Falciparum malaria is a deadly parasite for children under the age of 5 and predominantly throughout Africa. So that's the main killer, even though the mortality rate worldwide from all malarias is less than 1%. If you look at how many people are infected, it's an enormous number of people dying from them, and most of them are children, and most of them are Africans.
Have either of you had malaria?
No, I haven't.
I have not.
All right.
I used to raise that question, though, in the class. And invariably, we had a few students raise their hands, and later on I found out those are the Peace Corps workers.
Yeah, right.
Actually, on this case, one of the medical student that we sort of put primarily responsible, actually an MD PhD student here, I asked, how familiar are you with malaria? And he had malaria. He actually had grown up part of his life in Africa. So.
Yep. This episode is sponsored by CuriosityStream, the world's first ad-free nonfiction streaming service. They have over 1,500 titles. So if you watched one a day, that would take you 5 years down the pike.
You got to double up on the weekend.
Of course, they're always increasing, so I'm not sure you'd ever catch up. 600 hours of content.
That's a lot.
For people who like to binge watch,
Yeah.
founded by John Hendricks of Discovery Channel. So this means you're going to get real science, nonfiction, not reality TV. You can get it on the web or on any of the devices powered by the web, like Roku and Apple TV and many, many more. 196 countries. And they have science, technology, nature, history, interviews, lectures, lots of different topics. In fact, you can just go to the website, curiositystream.com, and look without having to sign up at all. You can see what they have. Things like Stephen Hawking's universe, where he traces the history of astronomical theories and technology. Always an interesting fella to listen to. Deep Time History, 3-part original on the 14 billion year history of the universe, which I've seen from day one.
How about?
Just wanted to see if you're listening.
I'm always listening.
No,
I might not be always looking, but I'm always listening.
Not true. Underwater Wonders of the National Park. It's a 7-part exclusive series.
That sounds interesting.
Takes you below the bodies of water somewhere Dickson has always wanted to be. But it's cool underneath those bodies of water. Right. This is the 100th year anniversary of the U.S. National Park Service. They also have one of the largest 4K libraries on the internet. 4K meaning ultra-high definition. Over 50 hours. They have monthly and annual plans starting at $2.99 a month, less than a cup of coffee. Check out curiositystream.com/microbe and use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That is 2 entire months free of one of the largest nonfiction 4K libraries around. Again, go to curiositystream.com/microbe and use the offer code microbe at sign up. We thank CuriosityStream for their support of TWiP, This Week in Parasitism. For your pleasure and education, we now have a paper which was published very recently in Cell. It's called the Genome-Wide CRISPR Screen in Toxoplasma Identifies Essential Apicomplexan Genes. Comes from principally Massachusetts Institute of Technology with a little help from UC Davis. That's the University of California.
I'm very familiar with it.
Davis.
They got a great ag school.
The first author is Saima Siddiq, and the last author is Sebastian Luridal. Yes, sir. So a CRISPR. You can use CRISPR.
That's where I put the lettuce in my refrigerator.
CRISPR is a technology discovered by scientists who are just pursuing their curiosity.
Right.
Originally found as a kind of immune system in bacteria and archaea with memory.
Indeed.
It has now become a tool, as we'll see in this paper, to manipulate genomes at will.
Right. I spent yesterday on YouTube looking at a number of visualizations of how CRISPR actually works, Cas-CRISPR system.
I'm glad to hear it.
It's fascinating.
I'm glad to hear it.
Fascinating.
Now, originally, the way it works in bacteria and archaea, let's say you get infected with a virus. The nucleic acid of the virus comes into the cell. There are nucleases that chop it up and then integrate pieces of the DNA into the genome into a specific locus. And then the next time the infection occurs, RNA made from that stored piece will then seek out the incoming virus, and together with a nuclease called Cas9, there are others, this is a very big and complicated system, but Cas9 is the nuclease that's been used, it will then chop up the incoming DNA.
That's amazing.
An immune system with memory. Amazing. Now, the people who were originally studying this, many people have studied it, there's a lot of controversy. Jennifer Doudna at Berkeley is one of the more prominent ones. She's done a lot of very important work. She said once, and she will probably get a Nobel Prize for this, I would bet.
Mm-hmm.
We'll see. Didn't happen this year.
No.
But we'll see. She said, when someone told me to work on this years ago, I said, why should I study this obscure thing in bacteria? Right? That's how science proceeds. Please. In fact, one of the Nobel Prizes this year went for the discovery of autophagy in yeast.
Right.
Another example of fundamental research that leads to big things.
Sure.
Now, in this country, we're moving farther and farther away from that, unfortunately. We want to cure diseases without doing fundamental research, and that doesn't happen. It's not happening. Take my word. But no one listens to me. Now,
That's not true. I listen to you all the time.
And then you laugh.
You have the Donald Trump disease. I can see they're sniffling.
I have a cold, yeah. It just came on this morning. It burns me up because I work with these viruses and probably.
Yeah.
Are there other viruses that can cause a mild upper tract infection?
There are many. Yes.
Yeah, there are rhinos and coronas and adenoviruses and paramyxoviruses, and I could have any of those. I should take a little of my secretions and do a little
Go down the
PCR,
hall.
but we don't have time for that. We have other we have other
And we're all
fish to
sitting
fry.
within 6 feet of Vincent.
We are.
So,
We're in the contagion zone.
yes, I'm a little.
Yeah, you could get infected, but I'm trying not to sneeze in your direction.
Thank you.
I did sneeze once. In this paper, they use CRISPR to disrupt 20,000 known genes in Toxoplasma,
Incredible.
one by one, using CRISPR, and then ask which one of them are essential or really important.
Right.
And then you would take them and study them, right?
That's amazing.
Is it 20,000 or 8,000?
I don't remember the number. Let's look it up. Yeah,
Because I think there's about 8 or 9,000. I think their genome is
human
smaller.
would be 20.
Yeah, so I think we're about 8 or 9
small?
for T. gondii. I think it's smaller.
Really?
I don't see a number in here, but probably
we'll get
one
to
of our
it,
listeners is
because
probably
I highlighted it.
researcher.
Well, let me
Sure.
scroll down here. It's actually possibly in the discussion, Dickson. Is that a possibility, Dickson?
It's
see that.
possible.
Here we go. 8,158 predicted protein coding genes. There you go.
Yeah.
Daniel, you were right. He's always listening.
He's always reading. That's your problem, Daniel. You're always
Now,
reading.
let's get some
reading.
technical
More comic books.
stuff on the table.
Yeah, please.
Toxoplasma, you can grow in culture. You have to grow it on cells, right?
Yes, you do. It's an obligate intracellular parasite.
You use human cells, right?
You do.
And what is the form that is multiplying in these human cells?
Tachyzoite.
Where do you get it? From other infected cells.
Exactly.
A tachyzoite.
Yep.
And when it
Freeze 'em up.
replicates in the cell, do they pop out at some point?
They do.
Like a virus.
They do.
And then they infect another
They
cell.
have a very viral-like life cycle.
They even say that they can see plaques formed by...
They can see the holes in the cell cultures.
Wow.
Created.
Can I make a wall of toxo?
I thought that would
if you would
would
like.
get you excited, Vincent, when they're doing plaque assays, right?
In fact, we have a new edition of our book coming out on parasitic diseases, and we've chosen as the cover, the poster child for the cover, a fibroblast.
He chose me.
A fibroblast infected with tachyzoites.
Yeah, you're right about that. When you finish your website, you can announce it here on TWiP.
We would love to do that. I think we just did that.
So what you do, there are two components of the CRISPR system. There's the nuclease,
Okay.
and they're the guide RNAs. So first you have to make a cell with the nuclease being produced in it. Okay? It's called Cas9. This nuclease not only can chop up DNA using RNA as a guide, the RNA will bring the
Find
protein.
the right gene.
The RNA hybridizes, and then the protein seeks out the guide RNA,
That's incredibly
Cuts it.
sophisticated.
and it will make a disruption, which will get repaired, and the guide RNA may get inserted, and it disrupts the gene. So now you don't have a functional gene anymore.
Right.
Now, what they did is take tachyzoites, is that,
Tachyzoites, yes.
and they introduced stably the Cas9 gene,
Mm-hmm.
and they didn't do well because they figured, well, maybe there's some random RNAs that are hitting targets, and even though we haven't put any guide RNAs in.
Right.
So they put in a decoy, and that's what made it work. This is a cool little
Yeah, I thought that was a
technical
neat
thing.
little technical.
They inserted, along with Cas9, a guide RNA that would target a non-coding region of a gene that they didn't think was essential. That way, in the untreated cells, without any guide RNAs added, this Cas9 wouldn't be chopping up toxogenes. It would go to this one. They would be distracted. You get what I mean, Dickson?
It's a diversionary tactic.
Precisely.
Yeah.
I thought that was brilliant. So they could make stable T. gondii with making Cas9.
Right.
Then, now usually if these are eukaryotic cells, you could make a lentiviral library of all the guide RNAs that you want to put in. You would have to have those synthesized. You would have to look at all the sequences of all the
That's
genes
a lot of
and
work.
pick maybe 10 different RNA sequences.
It's like a chip, making a DNA chip. Almost.
And have them synthesized, put them in your vector, and then you would infect. But they don't have any viruses of T. gondii.
Yeah, they do. There's actually a double-stranded virus.
But they're not.
Yeah,
They haven't
no.
been used
It's an
for
RNA virus.
gene therapy,
Yeah, I understand.
so they can't do it. So they have to introduce by DNA transformation.
Right.
And then you think about it. You have this library of thousands and thousands of DNAs encoding the guide RNA. And those guide RNAs are targeting individual genes in the toxo.
That's amazing.
Right? And they get in and they collaborate with Cas9 and they go to their gene and they chop and mutate it. And then basically what they do is they see what grows,
right,
and they sequence it, right, and they ask what genes are going away as they grow these several times in culture, right? They do several cycles. And the idea is that if a gene is essential, you're going to lose representation as it divides. If you've cut out a gene, it's going to lose representation as you grow them.
So how do you assay that part of it? Is that a hybridization technology that you extract the whole thing and then just hybridize?
They sequence it. No more hybridization.
Oh, my goodness.
Throw that away. It's in the museum.
Oh, okay. I'll happy to do so.
A French scientist once came. He was actually in the Goff lab, and he came in years ago, and I said, I'm sequencing by this method. He said, that's for the museum. This is how we do it now.
So the Pasteur.
So I love that. He said, this is for the museum.
That's very funny.
Yeah, they sequence. And that's why this is a big data paper. Mm-hmm. Lots and lots of data where they could look at the whole genome sequence as they grow this knockout library over time, and you can see what genes are going away. So in the end, they focus on 200 what they call fitness-conferring genes. Yeah. All right? So in other words, if you knock them out, and you grow the parasites, you like to kick out the deletion because it's hurting the parasite. And these are also lacking functional annotation. Do you know what that means, Dickson?
Not a clue.
It means the gene hasn't been studied with respect to its function. So if you go in the sequence of Toxoplasma, you can look at the sequence and they'll say it's annotated. This is a protein involved in DNA replication. This is a protein, blah, blah, blah. It's an enzyme. Okay. That's annotation. But you often have long open reading frames with no information.
I would have called those homology-like, but that's in the museum too?
It's in the museum, yeah.
Well, I think the homology would be if they said, oh, this is similar to something we've identified and annotated in another species, but the annotated is we've said,
actually studied
here, this one is studied, this is what it does, or what we think it does.
They would actually add that to the annotation. This is similar to a gene present here, and there the function is not known, or there the function is known, so it may be similar and so forth.
Right.
200 out of 8,000 that they worked on. This is quite a bit of work here. So many authors, as you know.
Yeah.
So one thing they did is then they could go back to wild-type tachyzoites and tag each of these 200 genes with a fluorescent label so they can see where it is in the cell.
Oh, my goodness.
So they can do that by CRISPR also. They can stick in a tag. Isn't that incredible?
Yeah, it is.
They don't want to disrupt it. They want to know, let's put a fluorescent tag in. Right. And there's this beautiful figure where they show where these 200 genes are located. You know, as you know, Dickson, these things grow in a vacuole, right, in the cell.
They grow in a very special vacuole.
What is it called?
The parasitophorus vacuole.
And there are multiple of the individual parasites, right? They make a little rosette. Would you call it a rosette?
It is a rosette, and they're all connected by the same cytoplasm.
It's amazing.
It is
until the final division stage.
It is beautiful, the symmetry. I mean, that's why we chose it for the cover.
That's right, exactly right.
Well, you should take one of these.
But we should have picked one of these with color. I mean, look how beautiful these are.
You know,
so they're labeling the gene products, and you can see.
But we couldn't afford that. I don't think we could afford it.
No, ours is a 2-color book.
So then they could say, where are they? Because these individual tachyzoites, are they called? That's not a tachyzoid, is it?
Those are tachyzoids.
In the rosette or tachyzoids?
Yeah, absolutely.
So could you disrupt them?
The only other stage that you're going to try to distinguish this from, they're called bradyzoites. And bradyzoites are in these large pseudocysts. Okay? Right. So they're totally morphologically different.
All right. So could you tease one of these out and put it on another cell? Would infect it? Would invade it and multiply and so forth? So these are fully infectious. Anyway, they can stain for each of these 200 proteins and see exactly where they are, which is really cool. And so they can tell, for example, that some are in secretory vessels. They have microneme-like structures. This is involved. You know what a microneme is?
Listen to him.
Do you know what that is?
I do.
Tell us about it.
Well, it's a secretory organelle inside the apicomplexan's nose, or the the apicomplex. That's the apicomplex. That's exactly where that is. That's
Isn't that right next to the rope trees?
Rope trees, yeah. Right next to the rope trees.
That's right.
Right. And
They have a lot of secretory granules also.
right up there at the tip, right?
Right at the tip. And they're secreted at the surface of the cell. Remember spit and kiss?
Yeah. These are effector molecules.
This is a John Boothroyd.
So anyway, some of these genes, the gene products are there.
Yep.
Some of them are in mitochondria. Some of them are in the nucleolus. The conoid is also part of this apical
That's
structure.
the very tip of it.
Right.
Exactly right. So protozoans are pretty complicated organisms.
Does it help you to know where it is?
Oh,
protein?
absolutely right.
That's your kind of thing, right?
Yeah, sure. I mean, what if it's a secretory protein? And yes, this is true. This is my kind of thing. You're absolutely
You like looking
right.
at pictures, right?
Yeah, but they can tell you a lot about. I mean, all these pictures that I see here of these stained versions of the tachyzoites with different proteins, it almost looks like you're looking at flowers. It's got a similar floral appearance, or some kind of a weird 4th of July celebration for each one of these things that bursts out of its cocoon, so to speak.
Every picture tells a story.
They're remarkable. That's why we have eyes to see with, Vincent.
That's right. And then they wanted to know what these, I understand, Dickson. They want to know what these genes are doing.
Well, that's yet another kettle of fish.
So, they re-disrupt them, actually.
They can tease them out.
And one of their initial measures of function was plaque formation.
How about
This makes me so happy. Because behind Dickson and Daniel is what we call the wall of polio. It's a stack of plaque plates.
Plaque.
One could make a wall of toxoplasma.
You could
right?
do that.
Of course, I'm not going to do it, but someone else could.
But John Boothroyd might have already done that.
No one would
like
be
him.
silly enough to do it as I have.
No, that's not silly
line.
at all. No, that's a life work in back of us here. You said that's not a lot of plaques, though, right? When I asked you how long it took you to do all of that, I thought it was years.
No, it was one experiment by a postdoc, actually. She did that over the course of maybe 1 or 2 months, yeah.
That's an amazing...
She did one big experiment.
Wow.
It was sort of like the experiment here. She had a number of genes. She wanted to know if they were involved in anti-polio activity, so she assayed them one by one.
Very cool.
So these studies, they find that a quarter of the genes are involved in invasion
Yes.
of the parasite into the cell.
Yes.
And that's not surprising. The parasite has to get in, right?
That's right. Absolutely.
So
well, it actually doesn't get into the cell.
It stays within a vessel.
This is where you guys had that fat. It's inside. It's within the cell, but it's within its own special.
Well, they call it invasion.
It originates.
Would you call it invasion?
Of course.
So if a car full of soldiers goes into the building, it's invading, but it may not be entry.
But they're still in the car.
Still in the car. That's right.
They don't get out
That's
of the
done every
car.
day.
Okay.
So the parasitopus vacuole is the car.
So the Trojan horse. Who was in the Trojan horse event, actually, in the old days?
The Greeks.
So were they actually not inside?
No, they never got in. They stayed inside.
Stop it.
A quarter of the genes. Are you surprised by that number? Of those 200?
No, actually, I'm not, because if you don't get in, you don't have an infection.
So it's important, and you need a complicated machinery to invade, obviously.
That's a fact.
You need a lot of different gene products. Of course, some of them may be redundant to some extent, I suppose.
This is true. So it would be interesting to know what the mechanism of plasma membrane invagination
Yeah. Well, now that we know the genes involved, they can start to study it if they can get funded.
If they can get funded.
But this being a parasite that infects almost all the world with no consequence, probably they're not going to get funded.
There's a lot of consequences.
I wouldn't say without.
A lot of consequences
I know, I'm riling you up.
No, I'm not easily riled.
No, I was going to say just last week we had a pretty serious infection with a gentleman that came in with this parasite in their brain in multiple different locations.
Right. It's a complication of HIV infection.
Right. By the way, they have a new name for these genes. They call them indispensable conserved apicomplexan proteins because they found them in other apicomplexans. Like?
Malaria.
Okay, what else?
Cryptosporidium.
Exactly. What does that cause?
Cryptosporidiosis.
Wow.
Diarrhea.
Diarrheal disease.
You have such knowledge.
It just...
They also find
I wrote the book on it. That was a joke.
Since sequences of other apicomplexan are known, they can see these eye caps.
Yeah.
It's the essential ones. They're present in the others.
These
as
are
well.
huge infections in chickens, too, by the way. The commercial poultry industry has a huge problem with these things. Tenella, particularly.
I think we talked about that. They're almost all infected. That's amazing.
Correct. That is correct. They spend a lot of money drugging out chickens for these diseases. Whereas if you had a vaccine, then you might be able to avoid all of that. So that's the promise.
They developed a nice system for studying the function of these genes by inducing their disruption. So you don't have continuous disruption, which we know is not good because these are essential genes. So they can add a drug and disrupt the function. And they show that at least one of these doesn't have an effect on the microneem secretion, but it has an effect on getting into the cell.
Right.
So they implicate these proteins in the events immediately preceding invasion of host cells. So that invasion process in the one example. This is probably where this work is headed. They've got all these genes now, and they can apply this approach, which is to knock down, they add a drug, and it knocks down the level of the gene product. And
they can get the whole sequence of entry this way.
Yeah, exactly. They could do that with each of these 200 genes if they would like.
Yep.
As they say, each gene is a PhD project.
It's actually really true. Yeah, breaking down each step.
Goff always used to do that. He would identify, by a screen, lots of genes, viral genes. Okay, here's your gene, go do your PhDs. And it works. It does work.
Having the entire genome sequenced makes a big difference here, too, I think.
Well, you wouldn't be able to design these silencer RNAs
without it. That's right.
Right, the ones that you put into cells to disrupt, you couldn't unless you have the gene sequence. So that's an important.
So people who are critical of spending a lot of money deciphering the genomes of these organisms don't realize that what you're really developing is a toolbox.
Of course. When they first announced that the human genome was going to be sequenced, this was like a $150 million project, which now in retrospect is nothing. This is how Craig Venter got famous.
Yeah.
This is over 15 years ago. I thought this was a waste of money. I really did. Because I thought we should spend it on, polio, for example.
And today?
Things. I think it was brilliant. It was
a fantastic idea. Right?
I was so short-sighted to think that. But that was very early on in my career, before I appreciated that you have to explore.
That's right.
And it's any genome sequence, you have to do it. It's absolutely important.
And without the tools, you can't explore.
Absolutely. So having CRISPR now allows you to do amazing things with the as they say in the last sentence of the first sentence of the last paragraph, genome-wide functional analyses have transformed the study of many organisms. You don't just look at one gene anymore. Like you studied a gene involved in angiogenesis, right?
Yeah, that's right.
You spent a lot of years on one gene.
We did.
Now you study hundreds of genes. Hundreds of genes.
Yeah, you could do it all.
Trichinella is not an apicomplexan, right?
No.
Do we have the genome sequence?
We do.
Can we grow it in culture?
No.
See, that's a problem.
Yep.
They can do this here with Toxo because you can grow it in cell culture.
I know.
Right?
And listen, we're painfully aware of that.
What do we grow it in?
You grow it in a mouse.
It's hard to do genetics in a mouse.
Yeah, but not impossible.
You could do a whole genome-wide CRISPR disruption and put them in mice and see what grows.
It would be challenging to get it into Trichinella because it's got an armor shell on the outside called a cuticle. So you have to electroporate them through using a shotgun.
Anyway, that's essentially it. Genome-wide screen. They've knocked out every gene, and they've identified a core of 200 that are essential.
Yeah.
They seem to be, in different parts of the cell. Some of them are involved in invasion, but others have other functions that remain to be seen.
Exactly.
Really nice story.
The beginning of a book on Toxoplasma.
Is this in your new book, this story? Probably it's too late. But this is the kind of thing you could add later, right?
Yeah, no, this is work done after we sort of put it together. So we'll have to see. Can we still squeeze it in?
We still have the galleys, and we're still working on them, so we could CRISPR it in.
Exactly.
All right. This is just one example of CRISPR. We are using CRISPR in my lab to identify a gene that's needed for a virus, and we have a cell line we've made stably producing Cas9.
Nice.
And then we put the guide RNA library into it, and then we infect, and we see the cells that survive, and those will be lacking the gene that's needed. It's cool, right?
Receptor molecule.
Receptor, or many other things, right?
Sure.
Now, let me ask you a question. I'm going to ask Dickson this because I love bugging him.
Okay.
I think I know what the answer is before you ask the question.
Let's say you knocked out a gene, and it's not only essential for the virus, but also for the cell. What would you see?
That's a lethal mutant.
Yeah, you would never know.
You would.
Because the cell wouldn't grow, and virus wouldn't grow either.
Would not.
So that's tough.
That is tough.
You will only find genes that the virus needs, but the cell can do without.
Correct. Or if you mutate the one in the cell, so it can still use it, but the virus can no longer use it.
You could, but these CRISPRs, they generally disrupt them.
Yep.
So you have what do we call that? When you disrupt the gene.
Site-specific mutagenesis?
Well, that's the general
And
term.
if you're going for knockout or
Knockout. Think in terms of the function.
Oh, loss of function.
Loss of function. That's
Loss of
exactly.
function.
This whole paper is about loss of function because you're knocking.
and we're allowed to do that.
Oh,
We
No
can
problem.
lose function. We just can't.
We're okay with
you
loss.
lose them, though. Where do they go?
The problem is the gain of function people don't like. But we shouldn't start there. I'll be an hour on that.
Yeah, please.
I get going.
Yeah, don't.
Okay.
Yeah, no, there's a lot of potential. I mean, I sort of joke that CRISPR is the new guitar. Like, there's always a joke, hey, everyone plays guitar. Now, everybody's
The new
using
guitar.
CRISPR.
Is it in
because
your lab?
it's-
Is it in the Goff lab?
Yes.
Of course.
It was in there, boom.
Boom.
Right away.
All right. I don't know if you have a case study so quickly, but let's ask.
Oh, I do.
Never a shortage.
I do. I have many waiting to come.
Dumbledore the elephant said, I'm all ears.
Since we do 25 episodes a year, you're going to need 25 cases a year. And if you did this for another 10 years, that would be 250.
That's a good amount.
Would you be able to have that?
I hope so.
Okay.
Because
The
you know
answer
what,
is unfortunately yes.
Yeah,
Because those organisms are still out there.
Yeah, they keep coming. They keep coming. So people keep coming.
And the same ones show up differently, depending on age groups and ethnicities. And
If medicine is successful, there won't be any more cases.
I mean, Mark Zuckerberg eliminating all diseases.
Is that what he wants to do?
Oh, I hope he does, but just with $3 billion, I think he's going to need more than that.
And especially if he limits it to California,
Exactly.
right? Because we know
Just gonna
here in
cure
New York,
diseases in California. That's
I think that's basically
like-
right.
here in New York is where the innovation
I don't mean to make
occurs, right?
fun of it because $3 billion is a lot of money to give to biomedical research.
Well, it's not enough, though.
But it can't cure all diseases.
But still, thank you.
Exactly.
I don't know if
Keep those dollars coming.
Mr. Zuckerberg listens to our podcast, but if he does, I think it's admirable what you're doing.
We do
People, the movers and shakers like Gates, Zuckerberg, Warren Buffett, who else? Anyway, they don't listen.
Howard Hughes.
They don't listen.
They don't have spare time. I don't know what they do with it, but
We don't have any billionaires that listen to.
You know that the
TWiP.
New York Times now has in the arts and entertainment. They have reviews. Now they've added podcast reviews.
Really?
Yeah, so they had a review of a podcast in California, which is of course by a Hollywood person, who's famous to begin with.
Yeah, well.
Why don't they do one of our podcasts?
I don't know.
Maybe they don't like it.
No, maybe they don't even know.
Maybe they don't understand.
Let's send them a link.
Who?
Whoever reviews
Them.
the Times.
Them. Okay.
All right, so let me give you. Now, this is a little bit of a twist. This is a family.
An entire family.
I'm calling this is entitled A Family with Eosinophilia.
Wow.
And this story was actually conveyed to me by I will say a good friend, a colleague, an infectious disease physician. Don't worry, you can't see anything over here, Dickson.
I used to do that with my wife when I played cards. She used to hold her hands straight down, and I lift them up so I can't see the cards, at least, for gosh sakes.
And so this is an infectious disease physician. And I have to admit, when she first started telling me the story, it actually took me quite a while before I believed that this was true. But you know what? She is smarter than I am. And no, I saw cases with her down in South America, and I was like, how come you always know the answer? I'm beginning to feel stupid here. And she claimed, "Oh, I'm older than you, Daniel."
She knows the lab technician in charge of.
No, so here's the story. This takes place in Australia. So a 45-year-old man. This is the dad. He is having a problem with mild abdominal distension. Okay. And so as part of his evaluation, he goes in and he's seen, and he has a complete blood count, which demonstrates an eosinophil count of 10,500, with normal being up to 500. Okay?
Enormous.
Now, his 45-year-old wife, right? Marries a woman the same age. She reports that she's feeling fine, but she goes and she gets a complete blood count because of this abnormality in the husband. And her complete blood count shows that she has eosinophils of 5,200. Again, more than 10 times normal. So they're getting a little concerned. So they have 2 daughters. The daughters are age 17 and 19. They don't have any symptoms, but they get their blood drawn, and one daughter has an eosinophil count of 900, and the other has an eosinophil count that is normal.
That's normal.
Okay. Now, they start digging a little deeper. What's going on here? Now, there's a story that 1 week prior to the dad starting to have symptoms, the sister-in-law came for a visit, stayed, shared meals with them, and she developed severe diarrhea, abdominal bloating, discomfort, 15-pound weight loss over the following month. And, they communicate, and she gets her blood drawn, and she has an eosinophil count of 4,700.
Oh.
Now, this family, as mentioned, lives in Australia. They live in New South Wales. They try to get kind of an exposure history, and they get this story that they eat raw fish purchased at the local markets. Okay? The family members have not traveled overseas or out of their urban environment in the previous 2 years. There were no pets in the household, no homegrown foods or anything else that we get a history of. We do get, as mentioned, this local market food has been this raw fish. And I think it's important for me to note that these are human beings, not penguins. Some of our longtime listeners will get that. They go back and they want to find out if these people always had high eosinophils or is this new? Mm-hmm. And they're able to go back. There are previous labs available. And prior, eosinophils were normal. There was no elevated eosinophils. So this seems to be new compared to prior labs. I'm going to give you a little bit more. They go ahead and they do. The first thing they're concerned about is strongyloides, right? So they do a full workup for strongyloides, serologies, stool, multiple times. It's all negative for strongyloides. So here we have this family, multiple people with eosinophilia, abdominal bloating, one person with significant weight loss and diarrhea.
The dad just has abdominal distention.
Yeah, he was the one who went first.
That's abdominal distension, else, right?
And that's how we found out that there was this sister-in-law who actually was the most symptomatic.
And
15-pound weight loss, diarrhea.
1 week prior, she visited, right?
Yes.
No one else had diarrhea?
No.
Where did she live?
She actually lived in the New South Wales part of Australia.
Close to
that's
them.
a big
So she lived close by. It is, yes, it is.
That's where Sydney is Eric?
And actually, yeah, these cases were seen by a physician in Sydney.
In Sydney,
Australia.
They don't live in Sydney?
No, they live in
the burbs.
Yeah, but in the city. They're not living in
Wagga Wagga.
They don't know them have HIV, right?
No, nobody is HIV infected.
Any remarkable medical history?
No, you go through and there's no remarkable medical history. There's no, as we say, toxic habits. Sort of interesting. We are in Australia, so you get that stereotype. But no, there's no report of excessive.
They all eat raw fish?
That actually seems to be a common thread.
Even the one that's
the
normal?
sister-in-law and even the one who is normal. The daughter who has no symptoms and has no increased eosinophils.
And this fish that they eat, is it freshwater or saltwater fish?
Oh, that's a good question. We don't know that.
My guess would be if they ate barramundi, which is a common occurrence in Australia because they like it so much. They're farm-raised barramundi.
Oh, yeah?
It's a saltwater / freshwater fish. It's both. So they have a lot of fish that are
Would a raised fish be less likely to give you an infection?
I would guess the opposite because you can concentrate lots of things in a in a monoculture.
So the other question then is, are there known outbreaks of farmed fish-borne diseases in humans?
There are, but they're mostly trematodes from Southeast Asia and South Asia.
Mm-hmm. As we've been speaking about.
Yeah, Clonorchus. I mean, we discussed Ophosorchus vivarini the last time we were talking. And so that's one example of a fish-farmed situation which could actually increase the occurrence of a parasite because it's so close to human habitation and agricultural runoff. And the agricultural situation is where they use human feces as a fertilizer for the eggs, that sort of thing. So, but they don't do a lot of that in Australia. And they certainly don't raise barramundi that way because barramundi need to feed on living other little fish because they're predators. Whereas the carp
So why couldn't this just be a fluke of some kind?
Why couldn't it be?
Yeah.
No, I'm not saying it couldn't, but the chances are that it's not because Australia is not noted for their fluke infections.
Okay.
But it's interesting because it poses the question. We can ask more questions.
Oh,
right?
please ask away.
Has this family ever gone on vacation together? And if they have recently to a place, for instance?
No, I think as we mentioned, it's been a couple of years since they've really traveled anewhere.
So it's probable, although this is just...
Oh, and the sister-in-law didn't go with them on this.
None of this happened before she arrived, though. That's the point.
Yeah, no, that's an interesting thing. Yep.
So whatever is happened after she arrived.
Yeah, so she arrives, everybody's healthy. And then a week later, right, the dad starts having symptoms.
And so does she.
She starts having symptoms.
Yeah.
Her symptoms are lasting, as we know, for like a month, right? It's been a month of diarrhea, eosinophils are up.
Did she bring any house gifts with her? Like
No.
food items that she makes herself and brings as a...
No, she didn't. No.
Or did they go out to an unusual place because she arrived and they decided to go out and celebrate and have a meal at a service?
No, they didn't. They had all the meals they ate at home. They didn't go out to eat while she was there.
The sister, that's her name?
It's a sister-in-law.
Does she also eat raw fish?
Yes.
Okay. Is this on a weekly basis, this raw fish consumption, or monthly, or what?
Every day, all
Every day.
Apparently on a regular basis. And it was one common thing that they all ate.
I see.
I mean, you say raw fish and then you go, gee, that's unusual. Well, if you eat gravlax, and they do all throughout Scandinavia, that's not unusual. Or Gruyne herring, which is a pickled herring, that's not unusual. We have smoked salmon. It's not cooked. You might say, well, that's not raw exactly. But the fact is that it's not cooked. So.
Did you eat raw fish for lunch today?
I had sushi for lunch today, and the answer is yes. I had tuna.
Yeah. I had some raw fish this weekend.
I had raw tuna, so yes.
So if we get sick with eosinophilia,
yeah, but these are.
I offset it with avocado, though.
But these are all frozen, right? Probably.
Well,
ours has been snap frozen here in the U.S.
Liquid nitrogen.
I mean, sushi restaurants have nitrogen, liquid or dry ice for that, right?
Or liquid nitrogen. Some of them are very sophisticated. They have liquid nitrogen. So the answer is yes. But in Australia, I think they're aware of the problem down there, so they would do that in most places where they serve sushi. Mm-hmm. But so this is an interesting development.
Yeah.
Very interesting development.
Do you have any ideas, Dickson? You don't have to say them, but just say if you have an idea.
I mean, I have lots of ideas, but I wouldn't dare share them until I have more laboratory findings.
Just want to make sure. Sometimes you say, I got it, and sometimes you
I didn't
have
get this
it.
one. No, I don't.
But here you have some ideas.
I have ideas. I know,
but you're not completely in the dark.
I'm usually completely in the dark, but not right now.
Okay.
I mean, there's a glimmer of sunlight coming through.
Yeah, no, but as mentioned, like when I first heard this case, even then I get some laboratories. I'm still a little bit, really? And then there's the treatment, and I'm like,
Treatment,
So.
Because sometimes you can do presumptive treatments. And if it goes away afterwards, then it was probably one of those things. So, but you'll never know for sure.
Okay.
Right.
Let's move on.
Intrigue upon intrigue.
This episode is also sponsored by Drobo, the makers of storage solutions for your computer. They come in a variety of sizes, 5 drive bays, 8 drive bays, 12 drive bays. You simply plug them into your computer, and they use a technology. It's a RAID-like technology, but Drobo calls it beyond RAID because it has some different features. For example, the You can put different size drives into your Drobos. Usually in traditional RAID, you can't do that. They have to be all the same. And the company was founded by a gentleman who lost pictures of his honeymoon. The pictures were stored on a RAID 1 system that failed. Now, RAID 1 uses mirroring for data protection. The best he could do was to reconstruct the drive, but it slowly developed data corruption as the media failed, and these bad data were mirrored onto the other drives. So one file got corrupted, and because of the nature of RAID, it mirrored the corrupted
Ruined
file.
Everything. Ouch.
So he said, there's got to be a better way. So he decided to develop Drobo, and that's what we have today. He wanted to protect your user data. He wanted to protect against a single drive failure. And so if a drive fails in your Drobo, you simply pull it out and replace it with a new one. The data are restored. He wanted it to last forever, which means you have to be able to increase the capacity. So he figured out a way that you could, if you start with, say, a Drobo with 5 bays, and you put 5 2-terabyte drives, someday you could swap them out with 3 or 4 or 6 terabyte drives, and there would be no problem. It would happen automatically. And it should also be simple to use, such that Dickson Despommier, for example, could use it. In fact, we gave Dickson one so that he could use it, and he swears to this day that they're easy to use.
They are very easy.
They have lights on the front, right?
They do.
Are your lights all green?
All of them.
That means everything's okay.
That's
if
true.
it ever turns yellow, that means think about getting a new drive.
Exactly.
And if it turns red, you're out of luck. There's no more space, or a drive has failed.
Oh, you're right. One of those two things. That's right.
And then you have to replace the drive.
Yeah.
In fact, a solid red means you're out of space. A blinking red means the drive has failed. But no problem. You pull it out, you simply slip in a new one. You can get it at Amazon or Newegg or many other places. Amazing. I've had Drobos for years. I love them. I'm so happy that they're supporting TWiP. And if you want to buy one, I mean, this is great for your home, for photographs, for music, whatever. Screenplays, plays,
Parasite
novels,
case histories.
Parasite case histories, PowerPoints. They also make a network one that you could plug into your switch at home, and it would be accessible throughout your house. Or you can use it in your lab. That's what we're doing here. You can go to drobostore.com and use the discount code microbe100. You will get $100 off your purchase of a 5-bay, an 8-bay, or a 12-bay system. We thank Drobo for their support of TWiP. And finally, as Harvey would say, John Harvey, was that his name?
No, Paul.
You have to say next to the mic.
Paul Harvey here.
Page 4.
Page 4?
Alex, we have a few email. Alex writes, hello, Vincent,
Vixen?
Vincent, Dickson,
That's
and
a
Daniel.
hybrid.
I am significantly behind on TWiP, so I can't comment on any of the recent case studies, but I have a good excuse that you are all complicit in. Let me explain. I majored in evolution and ecology in 2004 from UC Davis, which included a parasitology course that was one of my favorite classes. I rediscovered my love of parasites about 3 years ago, and shortly thereafter found TWiP.
Right.
You three helped me relearn the life cycles and non-infectiously exposed me to new parasites. The podcast helped motivate me to apply for graduate school.
Excellent.
To make a long story short, I'm now a new PhD student in the new disease ecology program in the Odum School of Ecology at the University of Georgia.
That's a fantastic place.
I was extremely busy applying for grad school, and now I'm extremely busy being a grad student, hence the falling behind. But it brings me joy that I'm in an environment where I can talk about parasites to people without any discomforting looks.
Very cool.
I'm taking their new disease course, which has included fascinating lectures on placental malaria and resistance to anti-helminthics.
Neat.
Of course, being a grad student has also been stressful, but at the same time, I've been happier. I'm hoping to do work on wildlife helminths in community ecology, but research topics are open right now, of course. I have many people to thank, but TWiP played no small role, so thank you. I've attached a photo of the shelf above my grad school desk, which I'm sure Dickson will appreciate. Did you see the photo?
I do see that photo.
Is your book there?
Our book.
You have to talk into the mic, Dickson.
I'm looking. Yes, it's there. I love those books. I love all those books he's got. Yeah, he's covering the waterfront.
So all I can say, Alex, I think this is great. Please tell others about TWiP.
Absolutely.
Please tell your parasite colleagues that they should listen. And they're going to say, I don't want to listen to a podcast. Just be firm. Get them to listen. We need more.
Tell them it's fun.
More listeners, because we want people to learn about this cool stuff.
Right.
Dickson.
Yes.
Next one. Anthony.
Okay, Anthony writes, Professor Despommier noted that the snail vectors of liver flukes are not native to the U.S. Are any of these invasive species in Florida? Are these Asian snails imported for aquariums, or do they live on plants brought in for the pet trade? If so, then the tropical snails and their parasites are very likely crawling about in Florida's waters. Have manatees been checked for exotic parasites? Well, those are two separate questions, of course. So the answer is it's possible to introduce exotic snail species into the United States, of course. One wonders why Biamphilaria glabrata, for instance, hasn't come from Puerto Rico, which isn't very far away from Florida, and brought into the United States for whatever reasons, contaminations on bird feed, that sort of thing. The eggs of snails go all over the world like that. I don't know why, but we just don't have a good record of any of the typical intermediate snail hosts for a lot of these flukes, and there aren't that many in the Western Hemisphere to worry about to begin with. So I would not be too worried about invading snail species in Florida. I would much rather fear the Burmese pythons and the Oscars in the rivers and that sort of thing. Florida is an open book, by the way, for that sort of thing.
I thought we did.
I don't know.
I thought we did a TWiP where we talked about the invasive snails being imported into Florida. Do you remember that?
I have a feeling those were land snails.
As
there was a land snail
to-
in Hawaii that we talked about as well, I think.
I know these were coming from Asia. They were imported for the pet industry. I don't remember if they were snails or...
Remember, you need two things. You need several things.
Yeah.
You need human feces that have the stages of the parasites, which will then be released into the environment.
Mm-hmm.
So the reason why, even if we had the snails, we wouldn't have the parasites is because we have a very good sanitary set of systems in most municipalities, which would prevent the exposure of the snails to these stages, which in most other countries that don't have good sanitation would not be able to prevent. So that's one answer to the question, why don't we have exotic parasites in the United States? Right. For instance, we have Anopheline mosquitoes all over the place, but we don't have malaria. And the reason for that, of course, is that every time we have a malaria case, they're brought to the hospital and treated rather than left outside for the mosquitoes to bite on them and transfer it to other people. So,
well, Anthony, check out some back numbers. I'm looking at the titles, and they don't help figure out what's in the episode.
Right. A manatee has been checked for exotic parasites. Well, those are not exactly people, and I wouldn't expect them to acquire the same kinds of parasites that humans do. So,
but they probably have their own.
They do probably have their own, and of course they're susceptible to lots of other non-exotic things like water pollution and boats running over them with their propellers and that sort of thing, and people hitting them with rakes. Believe it or not, they've actually been attacked by people standing on the bridges of Florida.
Nice.
Yeah,
They're so nice.
They're incredible. And then global climate change. In this case, it got too cold in Florida, and the manatees began to die for that reason as well.
Daniel, can you take the next one?
Star writes regarding pinworms. Thank you for your podcast. Although the info about not getting rid of them makes me sad. This is regarding pinworms. I am a mother of 3. My infant son, my middle child, and I all have them.
Oh, my.
I've treated my 11-year-old, although she has never shown signs. So there's nothing I can do to get rid of them. How did the wife manage them? You're a little quip or dismissive about having them. That is likely because you don't. Well, I think they're talking about you, Dickson.
Oh, dear.
The itching, sleep disruption, and irritability drives you crazy. Any suggestions are welcome.
Physician on call here. Come on.
No, so I will jump in on this.
Please do.
So, Star, let me tell you, Star, in general, what we recommend, so that I'm not actually treating you. But in general, this is an interesting issue. There are different ways that we can treat the pinworms, and our new book that's going to come out actually has a fairly expanded section on this, because it was actually my wife who commented about this, and I think it was certain areas like head lice and pinworms where people really want to know, what do I do?
Yeah.
and not just a medical person. Some of these therapies are over the counter.
Yeah.
And actually, the over the counter therapy, and I think I say so, parental palmoate. Palmoate, I'm pronouncing that.
That's okay.
I think it's called Pinex. I can pronounce that.
Right.
Pinex. And so what this is a single dose treatment. And you go ahead and you take this, and then 2 weeks later you take a second dose. But here are all the challenges, is that these little eggs, right? So we're going to talk a little about pinworm here. Is, you've seen all these documentaries about all these wonderful animals out there, but the pinworm is maybe the most incredible. The mother pinworm is such a self-sacrificing organism. She comes, and what she does is to lay her eggs. She actually everts her uterus, dying in the process, and showering, let's say, about 10,000 eggs up to 2 or 3 feet away. So these little eggs are everywhere, and they can actually survive for 2 to 3 weeks. So one single female can create this just mass of eggs. And here's the problem, is one person has them, and then the other person has them, and then they're all over the house. So it can often go back and forth from, the one child to the other child to the other child to the mom. And it can. I mean, there's a pruritus, there's an itching in the middle of the night, and this cycle can repeat over and over again. So a couple things that we recommend. One is this blind treatment of everybody, and then a blind retreatment 2 weeks later. But then there's this massive cleaning to try, because they're going to last for 2 to 3 weeks. So do you then even treat another time? Now, the nice thing about the Pinex treatment is you're talking about a $10 or $15 per person treated. I recently had this issue where a person that I know had a pinworm seam, are you ready for this, on colonoscopy. And they were prescribed one of the bendazoles.
Wow.
And it ended up being, I think it was $400. It is amazing what these medications, which are part of these multi-drug programs throughout the world, they're charging, $100, $200 a pill, and you have to take 2 pills, and then 2 weeks later you take another 2 pills. Wow. So what do you pay? $800.
Sounds like the EpiPen.
Yeah, no, it's really so I understand it's very difficult. It can be very frustrating to have these, and then so here I'm going to sort of throw in everything. So you've got this exposure, and from the time that you've been exposed, it's 4 to 6 weeks till later. So let's say you're exposed now, you go through the massive treatment, everybody's been treated, but now 4 to 6 weeks later, an adult female can form and the cycle can start again. So this can be quite a challenge. This is a really well-adapted worm, and so it may be frequent treatments and a lot of cleaning up.
So this Pinex, is this over the counter?
It's over the counter. You can go to CVS, you can go to Walmart. Yeah, it doesn't require
a lot of daycare centers have this as a problem too, because the kids pick it up at the daycare center and then they bring it home, and then it's and then it's hard to get rid of, and then they finally do, and then they go back and catch it again. So there's no immunity from this whatsoever, because it doesn't live next to the tissues. It actually lives in the lumen of the large intestine.
All right.
Yeah, so it's tough. So
we haven't given them any positive information here.
Well, no, I would say Pinex, just repeated treatments, lots of cleaning
everybody,
up, and I apologize for Dickson's dismissiveness.
Dickson, how can you be so dismissive?
It's just my nature, I guess.
We have one more from Wink.
I was going to say, what do you mean by that?
No. One more from Wink. Dear TWiP professors, here's a weird one. If you have the time, I'd love to hear if you have any hypotheses on the mechanism. He sends a paper in Clinical Infectious Diseases entitled Plasmodium Parasitemia Associated with Increased Survival in Ebola Virus-Infected Patients. In a cohort of Ebola virus-infected patients from Monrovia, Liberia, we found that patients were 20% more likely to survive when Plasmodium species parasitemia was detected, even after controlling for Ebola viral load and age. Have either of you seen this paper? It was published in August this year.
I'm looking at it right now.
1,182 patients with laboratory confirmed Ebola, 52% case fatality rate. Okay. They were 20% more likely to survive when they had Plasmodium parasitemia. Survival rate of 83%.
Mm-hmm.
I mean, an obvious hypothesis would be that the protozoan stimulated some branch of the immune system that also had an effect on Ebola. But Because Plasmodium does upregulate interferon, by the way.
Yeah, which would be antiviral, of course.
It could be an antiviral.
Yeah. Or the other is, a lot of times when we have, like poor Vincent here suffering from his virus, a lot of the symptoms, a lot of the disease is the immune system.
Sure.
And so, here's someone who's, maybe in an area where maybe the nutrition isn't great. They've got a Plasmodium infection. Once you treat that Plasmodium infection and allow their robust immune system to respond to the virus, that robust response may be what is a problem. So I don't know. I think there's a lot of interesting theories on why this might be true.
It would have to be explored in some kind of a model. First, one thing I would like to know is if they did cytokine profiles on the two groups, you see any differences that might be protective, right?
Yeah.
Right, absolutely.
I don't know if they had specimens to be able to do that. But let's take a look at the discussion.
They had blood samples that they used in this treatment. So the idea of going back and doing cytokine profiles on these 1,182 patients would be really interesting. Are you're seeing differences in the gamma interferon levels? Maybe higher levels of something else, or is it just lower? You're just getting a less robust immune response.
They say there are other examples of where Plasmodium infection protects against other infections, including Salmonella. Presence of Plasmodium can turn a self-limiting infection into a life-threatening one. Well, that's the opposite. And they talk about co-infection studies of mice. They suggest NK cells, induction of NK cells by Plasmodium might play a role. So this is something that you could study in animals, and they say they do have a cynomolgus macaque model that they could do this in. But the numbers look good. They have a lot of specimens, so I think statistically the significance is substantial.
I wouldn't recommend that as a cure for Ebola, though.
No.
Because of its high mortality rate.
On its own, right?
Yeah, exactly. Yeah, imagine the double hit on a kid that's only 5 years old. I mean, would be devastating, I think. And by the way, what age group was protected best? Does it say?
Well, that's a good question. Let's look. Let's go back to the videotape.
Indeed.
Okay.
Warner Wolf.
They had 1,182 patients.
They say independent of viral load and age.
Yeah, so all
age. Okay.
Effect was independent of treatment with antimalarials. Treatment with antimalarials did not affect survival in a mouse model.
Because all these people, as we see, were treated for malaria.
Yeah.
Right.
So they do have a mouse model. All of a sudden it pops up here. Let's go back to the paper and see where the mouse model is coming from. If they have data. 6-week-old mice were inoculated with Ebola and then malaria. Yeah. So is it validated in mice? Patients, mice. I don't see any mouse experiments here. Here we go. Effective antimalarial treatments.
Yeah, this figure 3 is where they do the mice.
Mice were inoculated with Ebola and then treated with antimalarials, but they didn't do a dual infection, which you would like to know, because I would suspect if you treat people who are malaria infected with an antimalarial, that may take away the benefit for Ebola, right?
Yeah, I think what they were worried here is people were saying, oh, well, you see the people, maybe there's something helpful about the anti-malaria meds. Maybe they have an Ebola benefit. That's right. So they wanted to show here that the malaria medicine wasn't...
It's not just the medicine itself.
Yeah, it wasn't the medicine. It's something about the malaria.
Good. Interesting observation.
Yeah.
Another TWiP crossover, Dickson.
I tell you, it's all connected.
You can find TWiP at iTunes and also at microbe.tv/TWiP. Please think about becoming a patron of TWiP and all the other Microbe TV family of science shows. You can find out more at microbe.tv/contribute. You can send us your questions and comments and try your hand at the case studies. It's really not hard to do, and we don't make fun of you if you get it wrong, because we get it wrong.
It's not.
TWiP at microbe.tv. Send your emails and your case guesses. Daniel Griffin is here at Columbia University Medical Center. Thank you, Daniel.
Pleasure as always.
And Dickson Despommier is at Racaniello.org, thelivingriver.org. Thank you, Dickson.
You're welcome, Vincent.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. I want to thank the sponsors of this episode, CuriosityStream and Drobo. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.